Hey everyone, thank you for joining today. My name is Wilhelm Huck. I'm a professor of chemistry at Radboud University in Nijmegen in the Netherlands. And our lab is part of the Building a Synthetic Cell consortium that we have in the Netherlands called the 𝗫𝗁𝗍 consortium, where chemists, biologists, a lot of physicists as well, and theoretical scientists together try and build a synthetic cell from the bottom up. And this is a 10-year project. We're a little bit over halfway, and we're making good progress. And today it's my great pleasure to introduce Roel Maas to you, who has been in BASIC right from the beginning. He is about to write up his PhD thesis, and he is going to share his result on, well, I guess I'll let him talk about the precise topic, but it's about an important aspect of building a synthetic cell. Roel, the Zoom is yours, and you can start sharing your screen.
Yes, I guess you can all see my screen right now.
Yes, that looks good.
Okay, perfect. And then let me just get started. So welcome everyone, and I want to thank you already for joining my talk. Like Wilhelm said, I'm Roel, and I'm doing my PhD in the group of Wilhelm Huck. And today I'm gonna talk to you about the work I've been involved in the last 4 years, and it's about a microfluidic optimal experimental design platform for the forward design of cell-free gene networks. So Wilhelm already described it a little bit, but we are part of BASIC, which is an acronym for Building a Synthetic Cell. And it's a consortium which consists of 17 different groups from 6 different institutes, research institutes in the Netherlands, which all work together to build a synthetic cell. Of course, not every group is making the cell on their own, but we're all working together on different different modules, basically. And at the end, we want to put all of these different modules together to build a synthetic cell. A couple of those modules are, for example, duplication. Of course, we need DNA, it has to be duplicated, division, but also membrane proteins for transport. Of course, also transcription and translation, because at the end, all of those different modules need proteins, which have to be transcribed from a synthetic DNA. And last but not least, we also, of course, need timing, because we need to time which of those modules is activated at the right, at which moment, because we first want the DNA to be duplicated before we start dividing our cell. So within the Huck group, we are focusing on 3 of the different modules. So we work on membrane protein insertion, and we also work on transcription and translation, where we mainly focus on the effect of the physical environment, like macromolecular crowding, on transcription and translation. This is beautiful work from a colleague of mine, 𝗕𝗦𝗆𝗵𝗆, which published this work 2 years ago already. And then also where I've been involved in is working with on the timing. So when this is done with synthetic genetic networks, and so we can learn how we can create those kind of networks to activate and inhibit the expression of specific genes time-wise. And this is also really nice work by Maruti, which we also worked on creating this kind of networks, studying the oscillators and getting a bit more information on how these oscillators actually work. So like I said, I've been mainly involved in my research around the question how to control gene expression in a synthetic cell. Like I said, we have all of the different modules, and we need to time the expression of all the proteins in the synthetic cell during the entire cell cycle, basically. So we need a set of modular genetic building blocks which can be used to either activate expression or inhibit expression. And we need a set of design rules to be able to, at the end, let me take a laser pointer, at the end, create networks to control the gene expression in the synthetic cells. And like I said, these networks are then called synthetic genetic networks. Of course, we're not the first one to work with the synthetic genetic networks. There has been quite a lot of work already put into this, and most of the work up to now has been done in closed systems, which we call batch systems. So those batch systems, they are closed. This means that there is a limited amount of nutrients in the system. But although it's a closed system, there has been a lot of work being put into this. And one of the pioneers in this field is the group of Vincent and Noreau, where they, for example, made this very nice cascade with a different set, with a set of different sigma factors, all activating each other, subsequently expressing 𝗲𝗎 at the end. This is all only activation elements, but of course, like I also said, we need inhibition also to shut down expression when we don't need it anymore. And for example, this studied in this work in a group of Richard Murray, where they looked at an incoherent feed-forward loop, which has direct activation, but also a delayed negative feedback loop. And there they showed this pulse-like response in a closed batch system, which you would expect for an incoherent feed-forward loop kind of system. Like I said, a lot of work has been done on batch systems, but it's kind of limited because after about 4 to 6 hours, you run out of the available nutrients in the system, or you run out of active ribosomes or RNA polymerases. So the interesting part only consists of 4 to 6 hours where you can really get good kinetic data out. However, when you would switch to a flow system, you would have continuous expression for over 20 or 30 hours, which and then the whole experiment can be used to extract nice proper kinetic data from. And so using this kind of flow systems, people have been already putting a lot of effort in studying more complex genetic systems where there's also protein turnover involved, like for example oscillations. So this is the work by the group of 𝗲𝗏𝗵, where they used specific, I think it was in glass cut out chambers with DNA brushes to study DNA oscillators, and also the group of Sebastian Merkel, where they used a flow system to look at ring oscillators like 3 and 5 node ring oscillators. So we have all of those nice examples. So what's stopping us from creating more and more larger and also more complex synthetic genetic networks? So one of the problems here is that we have all of those nice networks, and they provide us with a huge library already of different building blocks we can use to make those networks from. But the problem is that usually the characterization of those building blocks is quite difficult. And this makes it very difficult to forward engineer new networks from this library which is already present. And this is mostly because whenever you characterize a part in one specific system, the characterization is usually biased towards that system. So to improve the ability to forward engineer networks, we need to improve the characterization of those parts. And to do this, we first have to improve the data quality of the experiments, basically. So why is it so difficult to characterize these parts from synthetic genetic systems? So let me just show you this example. When we have the expression of just a single gene, and that you can kind of decouple the expression in a couple of processes. And of course, there is transcription of the DNA towards the RNA. Then you have translation from the RNA to the protein, and especially for fluorescent proteins, you have maturation, so the protein has to mature before it becomes fluorescent. Then we have degradation of the mRNA, and also, of course, regulation. So that's the transcription factors binding, which regulate the activation or the inhibition of the proteins. So usually the maturation rate is directly measured, but besides that, we have one control parameter, which is the amount of DNA you put in the system, and there's only one observable. That's the amount of 𝗲𝗎, which is produced at the end of the reaction. So there's all of those basically those 4 different processes, which all have one or multiple different parameters involved, and have to be estimated from a single observable, which usually means that those parameters are very collinear with each other, and so they can compensate for each other, which then results in identifiability of the parameters. And I think this is very nicely depicted here in this picture, where you just take a piece of paper, you crumble it up, throw it on the table, shine a lamp on it, and you cast this shadow. It just looks like a random shadow to us, but the underlying complexity of this shadow is actually a polar bear riding a turtle's back. So I think this very nicely shows that just having one single observation, it's very difficult to find the underlying complexity of the whole system from this single observation. So how can we then still improve the data quality without increasing the amount of observations in the system? So there's a couple of methods for this. And the first is going from a batch to a flow system, like I already said. when you work with a batch system, you have only 46 hours of expression when there is no limitation in the nutrients. So that actually gives you the kinetic data you want to retrieve from the system. However, when you're working with a flow system, there is steady state expression. So the whole experiment lasting up to 20 or 30 hours gives you information on the kinetics of the system. On top of that, you can use a technique called optimal experimental design, where you design an experiment in such a way that you increase the information content of that single experiment. So how we do this is, so how this works is that if you take two different parameters, so it's parameter 1 and parameter 2, and you take the error margin around those, the average values you estimated for these parameters, and those two form this confidence cloud. So this is just in 2 dimensions for just one ellipse, but you can imagine that if you go here, if you have multiple parameters, it becomes a cloud in multiple dimensions. So what you do during optimal experimental design, we try to minimize the surface area, in this case, or the volume of this cloud. And by doing so, you of course also then shrink the distributions of the parameter distributions. So you basically improve the parameter distribution, so you get them narrower, so hopefully also closer to the true value. And in computation terms, this is called optimizing the decoder of the system. And last but not least, you can also use like a term we call data-based fitting, where usually you take one system, you have a couple of experiments, but you fit one model to one system. This usually results in biasing the parameters of this system, of this model, towards that system. So when you have different systems and you put them all together with different experiments into one database, and you fit the whole database instead of one system or one experiment, there will be shared parameters between all those different systems, for example, which will break the bias of those parameters towards when you would fit them all individually. And on top of that, you can target specific parameters to estimate them better. For example, when you take twice the same construct, but you only take the parameters on the binding side, this would only have an effect on the translation parameters, whereas the transcription parameters or the regulation parameters, which will stay all the same. So in this way, you can target specific parameters to improve the estimation of those. So what we did, so we tried to combine all 3 of those different methods in one pipeline to see if this would really improve the parameter estimation. So at the end it came up to this. So we had to build some networks. So we made a small library to build some incoherent type 1-feed-forward loops, and we'll come back to that in a minute. Then using optimal experimental design, we created an optimal inflow pattern, which we can then use in microfluidic flow reactors to create a database of experiments. With this database, we then fitted this whole database all together to create hopefully very improved, really good parameter estimates, and to showcase that the parameter estimates are really good and are modular. And we try then to predict the behavior of some new networks. So we use the same library, but now it is fully characterized, and we make completely new networks which we haven't tested before, and we can see if we can predict the behavior on those networks based on the parameter estimated from different networks. So let's start. So at first we had to build a network. So we made a small library, and using this library, we made 6 variants of an incoherent type 1 feed-forward loop. So how this feed-forward loop works is that you have an activator, which is over here, activates the expression of a single reporter gene, while it also activates expression of a repressor, which then on its turn inhibits the reported gene. So we have a direct activation and an indirect delayed inhibition. So what we mean with the 6 different variants, so we used 3 different repressors in the mix. So we have a TED-R, PHLF, and SIMR. And for all of those 3 repressors, we made a construct either with or without a Radboud sequence. And for you now, it's enough to know that this Radboud sequence only affects the translation rates. So this is kind of this targeted parameter approach, which I've been talking about. So of course, on top of that, we also had to design a model. So what we did, so we took the transcription and translation process and we divided it in 4 different categories. That's again transcription, translation, degradation, and regulation. So we used the ordinary differential equations to model all those processes. And within the model, we had some specific assumptions. So we assumed that the transcription rate is dependent only on the sigma factor used. So whenever we have an operator site within the promoter sequence, that won't affect the transcription rate parameters. Like I already said, the Radboud J only implements the translation rates and a little bit also the mRNA degradation rates. There is no competition for ribosomes and core RNA polymerase in our system. And last but not least, their sigma 70 is present in the lysate and is present in saturating concentration. So we will never run out of that. So we could model that as a linear relationship, to transcription using sigma 70 as a linear relationship. Then we come to the microfluidic flow reactors we have been using. So those reactors are based on the reactors designed in a group of Sebastian Merkel by 𝗕𝗏𝗺𝗵𝗿. I just saw she's also in the audience. So thank you for making this beautiful reactor. So we tweak them a little bit, but how they mainly work is that so the reactor contains out of 2 different layers. So you have a flow layer and a control layer. So using this control layer, we can control the flow in the flow layer, and that works like this. So whenever there's an intersection between a control line and a flow layer line, when you pressurize the control layer, it kind of bulges up a little bit of PDMS, which is in between those 2 layers, bulges it upwards, and that results that the flow in this flow layer in that specific lane is stopped. And using this, we can control the flow throughout the device, and then there's 8 parallel reactors, and we can all control them individually. So we can do 8 different reactions at the same time if we want. And the device looks a bit like this when it's all plugged in and ready to go. So it's mounted on a microscope, so we can image the amount of GFP or mChRI that's produced inside of the reactors. So so now we come to the actual optimal experimental design. So what we need before we can do an optimal experimental design, we need some initial parameter estimates for all the different parameters we have to estimate. So to get those, we started off with a so-called step experiment, where we have the reporter constraint continuously flown into the reactor, and then over time we add the activator and the repressor, and then we remove the repressor and we move the activated DNA constructs from the system. And then the output looks a bit like this. So at first there was no expression because there was no activator there. Then when we add the activator gene, the repressor goes up. When we add the repressor, the expression goes down again. And we remove the repressor, expression goes up, and then expression goes down when we remove the activator. And we did this for a couple of different repressor concentrations for all different feed-forward variants to come up with some initial parameters. Using as additional parameters, we use our optimal experimental design approach to produce this optimized inflow pattern. And I think you can all agree with me that this is a very chaotic pattern. So I don't think any of us could come up with this by themselves. So we were very much wondering what this would bring us at the end. But before I show you what the experimental outcome was of this, we first wanted to make sure that this approach would actually work, so that the identifiability of the system would improve. And with that, we mean that the collinearity between the parameters, so that the parameters can compensate for each other, yes or not, and how much they can compensate for each other, goes down. And to simulate this, we simulated a couple of experiments. So we simulated the batch experiment, normal step flow experiment, the optimized flow experiment, and we also simulated those experiments all for all the different variants, and I fitted them all together to see how this database fitting would help us with improving the identifiability of the system. So on the bottom, what you see is that the collinearity between the pairwise collinearity between the parameters, and so the redder the box, the more collinear the parameters are, so the worse the identifiability. So what you see is when you go from a batch to a flow, there's already a huge improvement in the identifiability. And when you can go from a normal flow experiment to an optimized flow experiment, onto the database fitting, we see further improvement in the identifiability. So this this showed us that we are convinced now that we can perform the experiments and it would actually work to at least improve on the parameter distributions. So we performed this optimized flow experiment. So we used the same optimized inflow pattern for all three feed-forward loops. And then this is the data with the 𝘰𝗵𝗿. So this is the data which comes out. And again, of course, when you put in a chaotic input pattern, you get out a chaotic output. So at first it looks like very chaotic, and what can you make out of this? But when we try to fit all the parameters to this, and then compare it to the parameter estimates we got out, we see an initial parameters, so just from the step experiments, we see huge improvements in the distribution. So what's shown here is we took all the distributions per category, so per transcription, translation, decodation, and regulation, and then we kind of average them out, the boundaries, and see how much they shrink, basically. So what you see here is that for the step experiments, for transcription, translation, and regulation, you see a huge improvement in the boundaries of around the average value for the estimate. So we see a huge improvement in the parameter distributions. However, for decodation, there was not so much of an improvement, but that can also be explained because we have no data at all on the amount of mRNA that is being present at any time in the reactor. So there is no quantitative mRNA studies being done here. So that actually makes a lot of sense that then it's very hard to estimate degradation parameters. So to zoom in a little bit on this, to look at the actual parameter distributions for some examples, we show here the parameter distributions for the dissociation constants for different repressors. So we see that for all of them, we see a very large improvement on the distribution. And we think this is because of the optimal experimental design, because what we're doing here, we're constantly changing the repressor DNA concentration, which means that the system continuously has to respond to differences in the repressor concentration. And at the end, this would, and the system responsiveness at the end is very much determined by by how quickly the DNA, by how quickly the system reacts to changes in the repressor concentration. So by continuously changing this, it's better to estimate the parameters using this approach. Then for the transcription parameters, we also see a huge improvement, and we think this is largely due to the sharing of the parameters, because all of the transcription parameters are shared between all different feed-forward groups we are testing. And at the end, also the translation parameters also improve a lot. And we think this is hugely because we do the experiments with and without the 𝘰𝗵𝗿. And this then kind of is this targeted approach where we can target specifically the translation parameters, and this helps us a lot in estimating those parameters precisely. So after we now characterized all the different parts, we now try to see if we could predict the behavior of some new networks when we would create them. So what we did, we started off with a still relatively simple network. So we made a so-called pulse decoder, which kind of works like this. So we have an activated gene over here, which activates the expression of 3 different repressors, which all repress each other, and a reported gene at the end. So the mathematical law of 3 times minus is still minus, also applies here. So when all the different repressors are present, there won't be any GFP expression. However, when we don't have pH11 in the system at the start of the experiment, there will be GFP expression because CMR is being expressed, which inhibits TETR production, so GFP will be produced. However, when over time we do add a little bit like the PHLF gene into the system, we only add it for a very short time period, and then we remove it again, then the expression will go up. When we add the construct, the expression will go down, wherever there is not enough of the PHLF being built up in the system to completely shut down the expression of GFP at the end of this cascade. So there will only be a little bump in the expression before it goes back to the state it was in before. However, when you increase the time of this pulse, at some point you will reach a threshold where the signal is kind of propagated through the system, and the 𝗕𝗏 expression will be shut down completely for some time, and of course at the end will always come back up. So we perform those experiments with different duration of this pulse basically, and that's shown over here in this figure. So the triangles and the crosses are the actual data points, and the shaded area are the predictions we got out with the characterized parameters, with the characterized parts. And we did this experiment with two different concentrations of this gene over here. So this actually, so what you see in the y-axis is A over B. So this is relative expression. So what you see is that when you have longer pulse duration, indeed the expression, this relative expression just drops, so which is also expected. But what's more astonishing is that we can actually predict this behavior quite accurately, so to say. So this really showed that we can qualitatively predict the propagation of a signal through a network we actually never tested as it is now. So this is actually we were very amazed by this results. Kind of to step up our game, we tried a different, more complex network, which is the bistable switch, which consists of, again, the same activator with two different repressors which repress each other, and each repressor also represses one specific reporter protein, so either 𝗕𝗏𝗆 or 𝗕𝗏𝗁. And how the system works, like it's in the name, it's a bistable switch, so it has two stable states. So for example, when it is in the 𝗕R state, 𝗕R represses the expression of 𝗕R, so 𝗕𝗏𝗁 will be produced. That's what's shown over here. So when we then increase shortly the concentration of this construct, 𝗕R will also be produced at some point and will take over. We'll start repressing 𝗕R, and because 𝗕R is then being shut down, 𝗕𝗏𝗆 production will come up. So you will either be in the MR state or in the 𝗕𝗏𝗆 state, and you can switch between those states by giving a short pulse, basically, of the other repressor, and it will stay in that state until the next pulse is given. So we will switch back to the other state. So we performed exactly this experiment, and what we noticed is that here again the solid line is the actual data, and the shaded area are the predictions, that we can get almost quantitative predictions of our system based on parameters which haven't been characterized using this system. And we were actually very much amazed by those results, because this shows that we were able to take parameters and take one system, estimate parameters using that system, then change the system both topologically, but also change the gene structure of those genes we used to estimate the building blocks, basically. And then after that, we were able to almost quantitatively predict the behavior of that system. So this brings me to the conclusion. So I hope that I've shown you that we were able to build this library, this relatively small library of genetic building blocks, that we then characterize this library using 6 different incoherent type 1 feed-forward loop by using a combination of flow devices, optimal experimental design, and database fitting. And that then with this now characterized library, we were able to predict the behavior of 2 different networks, both on a qualitative level as well as on a quantitative level. So I hope that I convinced you here that our pipeline can be used to improve the characterization of genetic parts, enabling the forward engineering of complex cell-free gene networks. And hopefully maybe much more later on, when we start really building up this library, making it larger and larger and larger. So we can maybe even go towards reverse engineering of networks. That will be, I think, even cooler. Of course, I stopped this talk not without thanking some people. Of course, at first I want to thank Professor Wilhelm Huck for giving me the opportunity to do my PhD in his lab. Besides, I haven't done this work all by myself, so I want to thank 𝗕𝗏𝗆𝗿𝗆, which is a colleague of mine, and he's the computational mastermind behind this, behind all of this work. So without Bob, this work would never have been. We would never have been able to actually do this. So Bob, thanks a lot for this. On top of that, I want to thank Igors and Frank for helping me with either the microfluidic setup, with the wafers, or with cloning and lysate stuff. And in the end, I want to thank the collaborators from Eindhoven, the group of 𝘕𝗏𝗺, and that's Arjan and Pascal mostly, for helping me with setting up the microfluidic system here in Nijmegen. And of course, the funding agency at NWL and BASIC, and the entire physical organic chemistry department, which we are part of. And last but not least, also of course you for being here at the moment and listening to my talk. And I hope you enjoyed it. And if you have any questions, feel free to ask them, and I will happily try to answer them.
Thank you, Roel. Wonderful presentation, very clear, I hope. Definitely an amazing piece of work that you've done during your PhD. Congratulations on that. We have already a couple of questions in the chat, so everyone who hasn't done so yet, please put them in the chat and I can read them out to Roel. I will start with a question from Miguel Gonzalez, who said, "This is a fascinating idea. What are the criteria for including an experiment in your library? Do you include failed experiments too, or does it need to be a threshold of it worked well enough to tell you something useful?"
So in principle, every experiment would give you information. So actually, I think maybe even failed experiments could provide you with more information than an experiment which worked, because when something has worked, it basically says that it is following what you think it would do, right? So if it's failed, maybe it gives you more information about things you didn't know about the system a priori. So I don't think there is much of a requirement for the system. Of course, the system has to react. So if you add, at some point, we also added too much of the repressor, and then in our case, and then the system is just basically dead. So the repressor kind of overrules everything, and then you have a non-responsive system, and that of course then doesn't give you any information at all. So there is some level of it worked yes or no, so there has to be a signal, of course, but on top of that, I don't think there is
It's good to qualify though that failed means it didn't work as planned, but of course if it really fails because something is leaking or the lysate is unfunctional, of course that doesn't help. Then there are two questions coming from Carla Barden and Brendan Mortege, and they're kind of similar. I will start with Carla Barden's, no, they're not similar, but they address a similar problem. I will start with Carla Barden's one. Did you consider using any protein other than GFP as reporter? Something with shorter maturation time or with catalytic amplification like luciferase?
We've also used 𝘊𝗵𝗁, but of course that's the same, it's a similar kind of protein. No, we didn't really use any catalytic amplification like for example luciferase. Also, I think it will only complicate things more because you have more parameters to use to work around, because of course luciferase also has a specific catalytic activity. So I think that will only complicate it more. And for the maturation time, it doesn't really matter that much because we measure that directly. So we have a fixed value for that. So having a shorter or longer maturation time at the end doesn't really matter, I think, for the amount of information you get out of the system, because that's a known parameter. So you exactly know how long it would take for maturation.
And so that's why the next question is, I think, spot on considering this answer, because Brendan Mortege asks, is there a model related reason for focusing on protein expression, or did you consider looking at mRNA levels as well?
Yes, we did consider a lot, looking at mRNA levels, and we tried, I think, 5 or 6 different approaches. But the problem is that in our system where we have continuous expression, like it's very difficult to measure the mRNA concentration. So there are different aptamers, molecular beacons, but they usually either don't work or lysate or are not really quantitative. And on top of that, because we also flow out material, so there's a continuous inflow and outflow of material, we also model that the mRNA levels will be really low in the system, so nanomolar regime, which actually again makes it more difficult to actually to quantitatively image the amount of mRNA in the system. But definitely, if you would be able to measure mRNA, that would be a huge improvement on the system.
Brandon, if you have ideas, please contact us, because it is an ongoing project that we definitely would like to do, because it gives us much more information on the system, of course. Anna-Marie Denford has another question, and she asks, in the pulse duration experiments, could you investigate other parameters like energy dependence or how temperature affects the network?
In principle, you could put everything. So we have everything. We have it now on a temperature control stage. So indeed, you can check if lowering or increasing the temperature, what that would do to the system. Energy dependence, yeah, of course, you can also try to mix in different amounts of melters or energy solution to the system and see how that affects. So in principle, I think the ideas are endless. You can put whatever in, and whatever changes, if it would result in a change in the signal, it will always give you out some information, I think.
I think the energy dependence is going to be tough, because that's not really part of the of your model at the moment, right? So that will be a tough one. So temperature on the equations, on the processes in your structure will work. Energy dependence will require to expand the model quite a bit, a bit beyond what we, I think, know how to do at the moment, I would say.
Exactly. Of course, yeah, you have to include it in the model to
yes.
get information out of it. Yeah.
Harold Muriel asks, thank you for a fascinating and informative talk. Could you speculate what is the predictive power of your model? Could you use it to predict behavior of unknown parts?
Yeah, so the problem with unknown parts is that you don't have any parameter estimates for them, right? So yeah, that's difficult, because we only predicted, so we only predicted things with parameters which we knew the parameters from the previous experiments. So I think with this system you can only predict the behavior of a system where you have already estimated the parts for upfront. Right? I think for unknown parts it will be very, very difficult, because you don't know anything about those parts. That's my guess.
I think I agree with you. Dreaming forward, I would imagine, though, that if we would be able to make a large library of parts and would have characterized that, then at some point, I would hope you can use machine learning maybe to find sort of predictions for unknown parts. But I do agree that it is quite far away. Yeah. Another question from Henrike Niederholt-Meyer. She says, love this approach, great work. Do you get absolute values out for repressor binding, or is it hidden because of different TL rates, for example?
So we in the system we have so we use direct parameters for the dissociation of the what's called the repressors. So we do model that kind of directly. But of course, it is always a little bit in collinear with the translation parameters. So the collinearity between those is we shrunk the collinearities using this approach, but it's never completely gone. So there will always be some compensation between those two parameters, but you try to minimize that as much as possible.
Thank you. Let's see. There's a quick question from Brendan Mortega, who already had a question, but this is a quick one, I think. Great talk. Do you need microfluidics, or would similar work be possible in other compartments like vesicles or 𝗀𝗏𝗵𝗿s?
Yeah, so like I said, the great thing about microfluidics is that there's an open system, so there's a much longer time frame for the experiment to work with. So that's especially handy when you want to do the optimal experimental design. But in principle, like also in 𝗀𝗏𝗵𝗿s or in vesicles, in principle, you should also be able to make it long lasting if you have inflow and outflow of material. So in principle, yes. I mean, the problem there is that you cannot change DNA concentrations over time as we can do in our system. So then the optimal experimental design with continuously doing different DNA concentrations is harder. On the other hand, it's maybe more, it's maybe easier to do high throughput with closed batch systems, where you can just have a microtiter plate with hundreds of thousands of different reactions, different conditions you can test simultaneously. So I think there are pros and cons about both options, but I think you can use basically everything as long as you create a ton amount of data. At the end, the database fitting will always give you information out. But of course, the fitting becomes more difficult if you have more experiments.
Thank you. Michael Heimann asked the question, thanks, Roel, can you map face portraits, i.e. surf along a null line? Why not use a Hadamard transform to set the pulse sequences?
Ooh, those are some difficult words in there. So maybe try to, I didn't really get what a Hadamard transform is. So we also tried for the bistable switch, we tried to find the actual bifurcation point with some experimental simulations. We get really close actually, but that's much more trickier, of course, because there you have to be very, very precise. You have to exactly get the translation rates and the Hill coefficients to exactly map this point. But we actually get very, very close to that. So I think that's what you mean with the null line. But yeah, but the Hadamard transform, I don't really know what it is. So that's.
Let me see. There's a new message coming. No, Miguel, if you put us in the right direction for the Hadamard transform, or contact Roel or me later on, that would be much appreciated, because I think we need a bit more background information on that question there. 𝘊𝗵𝗆𝗵𝗆 had a question. Would you be able to speculate how much your results are specific to the architecture and nature of the genetic network you study versus specific to the type of translation system? Would the same network behave similar in yeast extract, for example, or isn't there enough data yet to speculate?
Yeah, so I think also when you would choose remain with lysates of E. coli, for example, there will be differences because there is always a batch-to-batch variation. And at the moment, like with the library we have now, the amount of experiments, I don't think we have enough data to make the same predictions when we use this network in a different system or like a different extract or maybe impure, for example. But I think this method, when you would include more and more experiments and also start including experiments of different lysates, it maybe will be possible to also detect the differences between the lysates and kind of pinpoint specific areas where why one lysate is better than the other, or what's the difference between them. So I think at the end, it's just a matter of increasing the amount of experiments you do, amount of systems you check, increasing the size of the database. And with every new experiment you add to the database, you get more information, and you will start breaking more biases. So we're always getting more close to the true values for the parameters, I think. And of course, also maybe at the moment, the model we have is kind of a simple model. So we didn't include research, competition, or energy consumption of all the different parts. But whenever you, I think there's also always a fine balance between the complexity of the model and the amount of data you actually have. So if you increase the data, so if you increase the database size, you can also increase the complexity of the model along with it and get more and more information and going deeper to the mechanistic understanding of the system.
Quite clear. Kate says thank you. And Todd Lutz had a question. Did your repressors use only proteins, or could you use physical repression like temperature or light inducible and small molecule triggers? This is a fascinating system.
Yeah, in principle, you could do everything. So at the moment, we're also in the group, we're looking at the use of Towel switches, for example, to repress at the mRNA level. So they'll again get more information out of the system. But in principle, with almost all repressors have a specific ligand. So you can also add this ligand to again remove the repression. This also gives you again more information. So yeah, so again, it's the more experiments, the better, basically, for the whole system. But I think you can use everything to start repressing. Also, optogenetics would also be an option because it's already on the microscope. So you could just shine light and specific laser and activate transcription and activate repression or activation at specific time zones. That would also be really interesting. So then you have another control parameter than just inflow of DNA. You can also first flow in DNA and then activate repression using light, for example. That's also very interesting.
There is one more comment coming in from Daniela Bittencourt, who says, thank you, great talk. And I think we're reaching the end of the questions. Roel, on behalf of everybody watching, really nice talk, great platform that you have developed. And I would like to sort of invite everyone who liked the idea to follow up on Roel's suggestion that the more data we put into the database, the better the model will become. So please, if you feel like you would like to do some measurements or would like to set this up in your lab, contact us, and we would be happy to see what we can do together to expand that database and train the system better. With that, thank you all for watching, and we'll see you again in the next presentation. Good evening.
Thank you. Good evening, everyone.
Bye-bye.